Today we're going to start lesson 5.7. The essential question is, how is the genetic information used to code for the expression of traits? So we're talking about how do we take DNA and make proteins. So just a reminder from Unit 2, where we learned about proteins being an organic compound, they have a lot of important jobs, like protecting the cell, communicate, turning food into energy. They're involved in movement, and other proteins like hemoglobin help red blood cells carry oxygen. Those are just a few examples. Protein synthesis is the building or making proteins from instructions on DNA. So again, synthesis means to build. We're going to talk about a new nucleic acid that's closely related to DNA, and that is going to be RNA. RNA stands for ribonucleic acid. Its job is to act as kind of like a middleman in protein synthesis. You're going to hear this term central dogma of molecular biology, and that's really talking about protein synthesis, the building of proteins. And it's this idea that DNA is converted into RNA, and then that's converted into proteins. The structure of RNA is different than DNA, and so we're going to just outline some differences. The sugar in RNA is ribose instead of deoxyribose. RNA is generally single-stranded. DNA was double-stranded. RNA contains uracil instead of the nitrogen-based thymine. RNA still has cytosine, guanine, and adenine. So the letters found in RNA are going to be U, C, G, and A. So A is going to pair with uracil rather than thymine. There are 3 types of RNA that we're going to include in our lessons. It's messenger RNA, ribosomal RNA, and transfer RNA. And these are all your vocabulary words. So if we take a look, messenger RNA is responsible for carrying copies of instructions for assembling amino acids into proteins. Ribosomal RNA is made up of, it's what makes up the ribosomes. So proteins and ribosomal RNA make up of ribosomes. There's more details if you're interested. The ribosome is made up of actually 2 parts. It usually just appears as a dot on our cell models, but it's actually a large subunit and a smaller subunit. And finally, transfer RNA, or tRNA, is going to be what carries the amino acid to the ribosome to match it up correctly with the messenger RNA from the nucleus. So if we take a look at DNA and RNA as they go through protein synthesis, your genes are used to basically make this protein. So there's 2 steps: transcription, which is DNA to mRNA, and translation, which is mRNA to a protein. There are some details here that you're going to see on the next few slides that talk about this is taking place in the nucleus and how it happens, translation taking place in the ribosome. So we have this model here, and we're going to get lots of practice with this, but basically inside of the nucleus, 1 strand of the DNA is going to separate. So here's your 1 strand, here's the complementary strand. This will separate, and mRNA is going to be made. This enzyme, RNA polymerase, is going to be building the mRNA as it goes. So what I always encourage students to do is to kind of write this little key down on their paper. Even if it's the test and you get the test, just remember, when you're going from DNA to RNA, A pairs with U, T pairs with A, C pairs with G, and G goes with C. And kind of keeping this will help you. So in this case, T, if we go up to our key up here, T goes with A. So this is the corresponding mRNA strand that's made, A, U, G, U, C, C. That mRNA is in the nucleus. It's going to be able to fit and leave the nucleus through the nuclear pore. This is what it actually looks like if we're kind of looking at this and we have 2 video clips that I'll show in class where they'll show you this in real time, what's actually happening. If we get ready to go to the next step, we have to talk about the genetic code. if you can open your textbooks and you want to learn more about this, it's in 5.21, so chapter 5, section 21. And the genetic code is going to talk about a codon. A codon are 3 consecutive nucleotides on mRNA. So if we look here, this codon is AUG, and this codon is AAC. So it's important to know that this genetic code that we're going to be using on the next few days is universal. All living things use the same code. It's unambiguous, which means that each codon codes for only one amino acid. It's also redundant, which means most amino acids are encoded by more than one codon. So when we're taking a look at these 3 bases, that makes up one codon. So if we have 9 nucleotides in a row, so 3, 6, if we had 9 more, we'd actually have 3 codons. If we had 5 nucleotides in a row, we would have 5 codons. So if you had 60 nucleotides, how many codons would you have? This is the codon wheel. If we take a look, the AUG codon, so this is how it works. You start in the center, so there's A. The second letter is in the second ring, so A, U, and then G. That is methionine. It's known as the start codon. So you should pause the video now, and you should try to figure out what these codons code for. Okay, if you've restarted your video, UAC is tyrosine, CAG is glutamine, GAC is aspartic acid, and UGA is stop. So we are going to actually stop, and you're going to practice, which is called the challenge codon wheel practice, and you're going to work on that and get your answers checked.